Hello class, welcome to our second lab, and today we will study the phylum Plasmodiophyra mycota, the phylum Haemimycota, and we will learn as well about Haustoria. In the phylum Plasmodiophyra mycota, we will study the slime molds or yellowing of grasses, which is caused by Mucilago crustacea, Fusarium, and Fuligo. We also study clapboard disease, which is caused by Plasmodiophyra brassica, and powdery scab of potato, which is caused by Spongospora subterranea. In the phylum Haemimycota, we study soft rot of cucumber and watermelon, as well as stalk rot of corn. Those diseases are caused by Phydium, and we'll also study white rust, which is caused by Albugo candida, and downy mildew of basil, which is caused by Peronospora belvari. So let's get started with the phylum Plasmodiophyra mycota, and the disease, the yellowing of grasses, which is caused by slime molds. The pathogens that cause this disease are Mucilago, Fusarium, and Fuligo. It is important to note that these fungi are non-parasitic, so they do not penetrate the plant tissue, and they just support their reproductive structures on the leaves and stems of grasses. This also helps them to facilitate the dispersal. What we can see when we have the disease is a large number of pinhead-sized sporangia on the grass blades, and those sporangia are white, gray, or purplish brown. It is important to know as well that these symptoms usually disappear within 2 weeks. When these pathogens reproduce in the leaves of grasses, they shadow the leaf surface, and therefore they impede the sunlight to come into the leaf blade. It causes a reduction in the plant photosynthesis, and therefore that's why we see the yellowish leaves when this disease is present. That's why the name of the disease is yellowing of grasses. The second disease that we will study is clapboard disease. This disease is caused by Plasmodiophyra brassica, and it is one of the most damaging diseases in cruciferous crops, including canola, broccoli, and cauliflower. Once the disease is in one field, it can cause serious yield losses, and it can even impede the growing of any crop in one field. The most important symptom of the disease is the root galling, which is caused by hyperplasia and hypertrophy of the root cells, and it impedes the plant to take water and nutrients. Therefore, we can see wilting and stunting of the plants. In this first picture, we can see the root galling. Those are young galls in canola plants, and we can see the wilting in a cabbage crop. And here we can see how a healthy plant is, and in the right we have the stunting, which is caused by the disease. How is this disease cycle? At first, we have the resting spores, which can live for even 17 years in absence of a suitable host. So those resting spores are in soil, and once they perceive a host root exudates, they germinate into primary zoospores, which swims towards the root hairs of the plants. Then they penetrate and cause the primary infection. The primary infection of the disease is only present in the plant root hairs, and there we have the development of a primary plasmodium, which evolves to a zoosporangium, and this one releases the secondary zoospores. The secondary zoospores swims towards the cortical tissue of the roots, and once there they penetrate and they produce a secondary plasmodium, and in this secondary plasmodium new resting spores are produced. Those resting spores, once those resting spores are produced, we have they are released again into the soil, and there the pathogen life cycle continues. It is important to note that in this secondary part of the The secondary part of the pathogen life cycle is whenever we can see those root galls, and it happens about 7 weeks after the primary infection occurs. In this part, we have the root galls are produced due to two main phenomena. The first one is the hyperplasia, which is the increase in the number of cells. So the pathogen induces the root cells reproduction, multiplication, and we also have hypertrophy. In this case, we just have super big cells, and most of them are usually full of the resistant spores of the pathogen. Here's how an infected tissue looks like. In this part, we have healthy cells. We can see how is a normal root cell, what's the size they usually have, and here we have hypertrophied cells. They are super big and they have inside lots of resistant spores from the pathogen. Those purple things we see inside, those are the resistant spores of the pathogen. The third disease we're studying is powdery scab of potato, which is caused by Spongospora subterranea. We have two kinds of symptoms with this disease. We have the ones which are observed in the tuber, and the ones that we can see on the roots. In the tuber, at the very beginning, we have purplish brown lesions which evolve to small blisters, and at the end we have round open lesions like these ones in the picture, which exposes the masses of spore balls. That's why the name of the disease is scab. And in roots we have the same galling as we had in Plasmodiaphora, but this is caused by another pathogen. We know this disease as powdery scab because scab is an open lesion on the skin of the potato, and here we have a powder which contains large numbers of spore balls. This is pustule section of potato scab, and here we have the potato tuber, the cortex, the epidermis, and inside of it we have the spore balls. This is the spore balls which are among disintegrating cell walls, and this is a spore wall within a cell. This is a picture of how a spore ball looks like, and they are very spongy. You're going to see this during your guide and also in the lab quiz. Now we're going to change to the phylum Haomycota, and the first pathogen we're studying is Pythium. This pathogen causes different diseases such as soft rot in different plants, star rot of corn, and damping off as well. Damping off is a disease of seedlings, and when you see the seedlings that they just die at very young, very young. So here we have root rot caused by Pythium, fruit rot, stem rot, the typical damping off, which Pythium blight, and here we have soft rot in cucumber and in watermelon. The typical symptoms of this disease are soft, dark green water-soaked spots with humidity, and if there is free water, we're going to have the growth of cottony pseudomycelium, which is what we can see over here in the picture. Have you ever seen anything like this in your fridge? I think you have. So how's the disease cycle for the homomycetes? So to begin, we have an oospore, which is the sexual spore of the pathogen, which germinates and produces a sporangium. And this sporangium produces zoospores, which are released, and they produce seeds, which germinates, then form an apresorium, they infect the plant, and once it grows inter and intracellular, produce the inter and intracellular mycelium, it produces asexual spores which are produced within a sporangium, and these new asexual zoospores can produce as well a cyst that germinates and reinfects. So we have two different types of spores. The asexual ones, which are the zoospores, which are produced in the sporangium, and the sexual spore, which is the oospore, which is produced by fertilization of an oogonium by an antheridium. The oospores are double-walled and they look different to the sporangium. As we can see here, we have an oospore and they are double-walled, like all those over here, and we have sporangium over here and one over here. The other disease we're studying is the white rust, which is caused by Albugo candida, and this disease affects as well the Brassicaceae family. The typical symptoms are white blisters or pustules on the leaves, flowers, and stems. The blisters usually contain large numbers of sporangia. These are the photos of the symptoms. It also causes the stag head, which is the flower malformation, as we can see over here. And the disease cycle for this pathogen is almost the same I exposed before, but how those oospores are produced. So for an oospore to be produced, we need an oogonium, which is the female, and an antheridium, which is the male part. So, and they are originated from the mycelia in the intercellular spaces of the host tissue. We have here an oogonium that's kind of circular, and we have an antheridium which is kind of C-shaped. We have over here another picture. We can see inside an oospore the oogonium and the antheridium. There are three main stages in the production of an oospore. In the first one, an oogonium and an antheridium come together and the fertilization begins. In the second stage, we have the developing oospore. In this case, the fertilization has already occurred. We have the fertilization tube, as we can see in the picture. We have the oogonial wall, which are the remains of the oogonium once it was fertilized, and we have the developing oospore. We have this double wall being developed. And in the third stage, we have the oospore, which has already produced a dark, thick wall, and it is ornamented as well. With the oogonial wall, it's over here, and the fertilization tube has already disappeared. Plant pathogens can be of two types. We have the biotrophs, which are organisms that can live and multiply only on another living organism, and they can obtain their nutrients without killing the host. These biotroph organisms are those that we cannot grow in petri dishes, and we have necrotrophs, which are organisms that feed only on dead tissue, and they kill the whole cells and feed on dead cell content. They usually produce different enzymes to kill the host and be able to take the nutrients and feed themselves. It is important to note that the pathogens that we have studied today, all of them are biotrophs. Now we will learn about the Haustoria. Haustoria are simple or branch projections of hyphae that penetrates the host cell and absorb the nutrients. This is the part of the fungus that actually takes the host nutrients to feed itself. In Peronospora parasitica, which causes downy mildew, we have intercellular mycelium, which has large finger-shaped Haustoria, and we're going to see how they look like in the next slides. But what type of organisms do you think that Peronospora parasitica is? In the previous slide, I already gave you a clue. So are they necrotroph, biotroph, or saprophytes? Here we can see the haustorium, which is a finger-like structure within the cell. This is the cell. This is the intercellular space. And here we have an intercellular hypha. This is another picture with different magnification. Here we have the pith cells, the intercellular space, the haustorium, which is finger-like, the intercellular hypha, and we can see that all these small dots in the haustorium are finally nucleus. The last thing we're going to learn today is how to do a leaf imprint on agar. This is a methodology that we do just to study the resident microflora on leaves, and to do it we just lay a plant leaf on the surface of the medium to impress on the agar. We leave the leaf carefully and we just try not to touch the medium with our hands, and this is pretty much it. I invite you to watch the video about how to do a leaf imprint so you can better understand how we do it, why we do it, and what it is. So now we're done with the lecture. You just have to go to the link on eClass to complete your second lab quiz, and using all the information on the slides, the video, and the lab guide, go and answer the questions. Just a reminder, you will be able to complete the quiz until 1 hour before the next lab session, and you can go back and forth as many times as you want, and after completing the lab report, don't forget to click and submit, and enjoy your lab. Bye!